All right, everybody, here we are. This is the final lesson for the year. To all those people that have stuck it out, congratulations. There won't be much work attached to this. This is just for your information, some interesting stuff about the human eye and how it's able to process light. Yeah, let's get right to it. Okay, so this is part biology, part optics. We're going to talk a bit about the physiology of the eye, how the various structures work to process that light. So some of the stuff that we've learned in optics with regard to refraction, we'll go over the actual biology of the eye and how it works to refract that light. So if you know, take a look. You don't have to fill this stuff in. I'll give you a note that accompanies this that you can just kind of follow along with in terms of what all these structures are. So that blue structure there you see is the lens. The lens is one of the structures that is used for bending the light, refracting the light. The retina, that's the spot where the image is actually formed. So the light rays that are reflected off an object will come through and eventually focus on the retina, and you'll see an image. The first part that light will go through is a transparent portion called the cornea. So the cornea, as you can see, it's shaped such that it will bend the light before it gets to the lens, which will then have that light undergo a second bending before it reaches. And if it's done correctly, if the geometry is correct, the light rays will converge on the retina, producing an image. So I know we didn't have a lot of time in optics, but this is a convex shape. Same with your lens, it's convex. Concave would be the opposite. And convex lenses are converging lenses. So they will converge light rays to where they come together at some point. Whereas concave lenses are diverging lenses. They will take incoming light and cause that light to separate. You can't see my hands. Oh well. The light rays will go in opposite directions, never converging when they pass through concave lenses. There's the pupil. The pupil is just really that hole, that opening that allows the light to go from the cornea through. And of course, you've heard it can be controlled. We'll talk about these muscles in just a second, but it can be controlled, as you know that when there's low light conditions, your pupils dilate to allow more light through. When there are high light conditions, your pupils will contract because there's so much light that you don't need it all in order to produce an image. The iris, so that's the colored portion of the eye. I know we talked in biology a little bit about mutations that cause blue eyes in human beings, and we talked a little bit about dominant recessive traits with respect to brown eyes and blue eyes. That color in the eye is really the iris. So that's the colored portion that has that pigment, has the melanin in it that causes color of the eye. The sclera, that's the whites of the eyes. It's a very tough part of the eyeball. It helps maintain the shape of the eye and protects it from deflating in case of injury. The optic nerve is what connects the eye to the brain. So it connects the image that is produced on the retina to the brain where it is then processed. So that image is actually processed and you can make sense of it. We have, whoops, sorry, let me go back. Cornea, yeah, that's all, I guess. We'll talk a little bit about these pockets here, the aqueous humor and vitreous humor, later on. So if we're to break down the human visual system, we can kind of break it into four main categories. So those things that are necessary for refraction, for bending of the light, which include the cornea and the lens. Those things that control the light, how much light gets in, which is the pupil and the iris. Well, I got cut off a little, but light detection. So it's like the film in an old school film camera, and that would be the retina, where that light is actually detected and an image is formed. And then lastly, processing, again, cut off, and that's done in the brain. So the brain is responsible for processing that light that was detected on the retina and forming that image. So let's take a look at how that image is formed. So if I have an object here, this arrow off in the distance, I'm going to get light reflected from either, street lights or the sun or whatever the case may be. It's going to be reflected from that object, and it's going to first, let me go back and show you that again. Those light rays are going to hit the cornea, and the cornea will bend the light the most. So remember the object, the idea is to have that light focused on the retina at the back here. So the cornea is responsible for the most refraction. It bends it the most. Then it will go through the lens and bend again until it hits the retina. Take a look. Okay, so it bends once at the cornea, bends again at the lens, and then hits the retina. Here's the interesting thing. That image, if it's below this horizontal line that you see here, the image is actually going to be upside down. Let me show you that again. If it were above this line, the image would appear right side up. If it hits below this line, which that light ray does, the image is actually going to be upside down. So your brain is a wonderful thing. Even though the image that's actually produced is upside down on your retina, your brain almost instantaneously flips it right side up. So, because obviously it would be harder to do daily activities with everything appearing upside down, although maybe you get used to it. I don't know. So, is the world really upside down and your brain changes it? Hmm. Questions to ponder. All right, let's talk a little bit about the cornea. So, the cornea is this transparent part at the front. You can see how it's shaped in that convex style that's going to converge light rays, cause them to bend. So the outer wall of the eye, this hard white part, the whites of your eyes, that's the sclera. Okay, so that's that tough uh, outer portion. If we were in class, like at Beale, we would have got to do a sheep eye dissection where you would see that that sclera is actually very, very tough. And able to get through it, you really have to apply a fair bit of pressure, and that's good because you don't want an injury. So, if you hit your eye and for some reason, you don't want the sclera to easily break. If so, you're going to lose pressure in the eye and it's going to cause all sorts of problems. So it's very tough. The cornea is that transparent portion, that clear portion that lets light through. And the shape is very important. If you listened in whenever yesterday or the day before, I talked about people that are nearsighted, like Mr. Sanford. We have more than likely, there's a couple of reasons, but quite possibly misshapen eyeballs. And it's likely that that cornea might be too curved. It's too curved and it causes the light to bend too much. And that causes the image to be formed in front of the retina, which is why I need lenses to slow down that bending of the light. In fact, my lenses will actually cause the light to diverge before it then hits the cornea and then converge so that it creates an image right on the retina. Most refraction takes place here. Already said that. So more refraction takes place at the cornea than actually on the lens. The iris and the pupil are strictly for light control, right? They can open up. And as I said, the iris is the colored portion. So they can open up in low light conditions in order to let as much light through as possible. They will close, contract in high light conditions because you don't need as much coming through. And if you're a photographer, it's kind of like the aperture on a camera. So you can see those pupils are able to either dilate, this would be what happens when there's low light, or constrict when there's high light conditions. And these muscles that are attached to the lens here are very important because the lens can actually change shape. So that's what allows you to focus both far away, so off in the distance, or close up when I'm reading a book. I can do both of those because I can change the shape of my lens. I can do it less than you can. I'm getting older. You lose the ability to change the shape of that lens as you age, which is why older people Eventually, we'll need reading glasses, or in my case, I'll eventually need bifocals, one part to see close up, one part to see far away, because I lose that ability. My lens loses flexibility. So the lens is transparent and can change shape. And as I said, that allows you to both see, focus on images far away and then close up. I wouldn't try this for too long, but if you look off into the distance, you can look off, focus on something, 50 meters away or further as far as you can see, and then look at something really close up until you focus far away, close up, and those muscles will actually begin to hurt, just like if you were doing curls, because you're really working those ciliary muscles in order to get change the shape of the lens to see far away and then close up. Don't do that for too long; it'll hurt your eyes. Right, for close-up objects, the lens actually becomes thicker. For further away, it will become thinner, stretched by those ciliary muscles. Let's take a look at how that works. So the top diagram here is for an object far off in the distance. So in that case, the ciliary muscles are relaxed. And when they're relaxed, they're pulling on that lens. So it's thinner in this case. Oops, let me get this out of the way. This entire thing is called accommodation. So the process of allowing that lens to either get thinner or thicker is called accommodation. You're accommodating for both far away and close up so that you can focus on both. And when those muscles contract, the lens will actually become thicker. And yep, called accommodation. That's what I said. So let's see how that could work. So for an object far away, if the lens, if those muscles were contracted and the lens is thicker, it would actually produce an image in front of the retina. Remember, we want the image to be here. That's no good. So what has to happen is the muscles have to relax. And when they do, the lens gets thinner and the image is properly focused onto the retina. The opposite happens when we're looking at an object far away, the lens is thinner, the muscles are relaxed, and then we want to check something out. We want to read, a book or your phone or something like that. Well, those muscles will have to contract and make the lens thicker so that that image will now focus right on the retina where it should. I didn't mention these in the physiology portion where we looked at the labeled diagram of the eye, but there's a pocket in front between the cornea and the lens called the aqueous humor. And there's a pocket behind here, behind the lens and the retina, called the vitreous humor. And they're filled with fluid. And it's transparent, obviously, because you need light to go through. The aqueous humor is a little less viscous than the vitreous humor. And they can provide nutrients to the cornea and lens and also help maintain the shape of the eyeball. It's very important that the eyeball maintains its shape because any change in the actual geometry is going to cause those light rays to go in ways that you don't want. It's going to cause an image that's not clear, not focused on the retina. So it's very important that the shape is maintained. Which brings us to the retina. So that's like the film in an old school film camera. That's where the image focuses. And you have different kinds of detectors, different kind of light receptors. One are rods, and you might have heard of rods and cones. Cones are for color, rods are for black and white. Rods allow you to see light in low light conditions. So it's only black and white, but you only need a little bit of light for those rods to work. Whereas cones are for color, but you need much higher light conditions for the cones to work. So if you turn off the lights, but you got a little bit of light coming in, maybe from a window that's not fully sealed off by curtains or something like that, so you have a tiny bit of light coming in, it'll be really tough for you to distinguish a dark red shirt from a dark green shirt. Why? Well, because you won't have your cones working very well if there's not enough light, but your rods still will. So they'll see black and white, they'll see in shades. But if those shades are equally, if it's a equally dark red and dark green shirt, you won't be able to tell the red from green, but you'll be able to tell that they're both dark in color because those rods will be working, but the cones work. This is just kind of an interesting note that looks at biology and evolution while also looking at how your eye works. If you were to design an eye, here's something you would never do. So the spot here that's highlighted by the box here, I've blown it up. Here it is larger. You see the optic nerve. This is, remember, the connection from the eyeball to the brain so that your brain can process the image. Well, these nerves actually pass in front of these photoreceptors, the rods and cones from your retina that actually are there to process that light. So, in that spot right here where the optic nerve connects, we've got an issue. Light has to actually travel through this area, and it can't because the nerves are in front of the retina right here. So, you actually have a little blind spot right where this box is. What your brain does is it fills in the image. So imagine if you had a grid that had 99 white squares, but that one, so a 100 square grid and 99 of those squares were white, and you had one that you couldn't tell what it was because it was blocked. That's your blind spot. Your brain fills it in and says, if all the rest of those are white, that one's probably white too. So it fills it in, and so you don't see that blind spot. Obviously, it's a little more complicated than that because we don't just see in black and white squares. But if you see an image, and depending on the way the light is and where you're looking, the small portion that's blocked off by that blind spot, your brain just fills it in and says, well, whatever's around it is probably what's there too. Um, so it's not always true. If you look online, there are uh, several optical illusions whereby you can kind of trick your brain's filling it in when really it's different information that's there. So you can look up some optical illusions with the blind spot just to show you that that's true. Just to show that evolution is not this process of finding perfection. The reason that, so by the way, this doesn't occur in some other species. So, for example, octopus, octopi, octopuses, well, they don't have a blind spot. Why? Because their eye developed completely separately from our eye. And whereas this is just a series, remember, mutations occur, and if they're beneficial, they stay. If they're neutral, maybe they stay. If they're not beneficial, so they're not positive, and they cause a decrease in the fitness, meaning you're less likely to reach the age at which you pass on your genetics, you breed successfully, well, those traits get washed away. Well, this is one of those traits that's mostly neutral. It didn't provide a benefit, but it also didn't really, because most of the time your brain works pretty well to fill in that blind spot. It wasn't uh, bad enough, deleterious enough to get washed out of the population. So this mutation survived. It's not the ideal solution to process light. You don't want that blind spot, but that's not the way evolution works, right? So since it didn't cause a big enough change in fitness, it stayed. Whereas this particular mutation didn't occur in eyeballs of other species like the octopus, so they don't have it. So they actually have a better solution to this problem than we do. Why? Well, because that eye developed completely separately than ours. And that's all the stuff that I said. Uh, rods and cones. I'm not going to go over this too much, as I said. Rods for black and white, cones for color. I'm going to let you guys try this as you watch. It just fills in. If you keep blinking, you can kind of see little black dots appear at the intersection points. And again, that's your brain filling those in. If you want to see some more optical illusions, I'll let you click on those and then you can watch some more. Last thing I'll talk about are eye defects. So Here is a nearsighted um, versus regular versus a farsighted eye. So this is what should occur normally. You get that image perfectly, those light rays converging perfectly on the retina. If your eyeball is shaped funny or your cornea is too curved, what you have is those light rays converging in front of the eye. That's called nearsightedness or myopia. Likewise, you can have a problem where if your cornea is not shaped, not curved enough, the object will actually be in focus behind the retina. Obviously, that can occur. So that's called far-sightedness. It's fixed. So nearsightedness is fixed by putting, so in nearsightedness, you can actually see close up. So I can see, right here, okay, I can read a book like that, but I just can't see far away. So you need to fix it by putting a concave lens in front, which causes the light to bend first before it hits my cornea, and then eventually converge on the retina so I can see clearly. Whereas the opposite for farsightedness, so hyperopia is farsightedness, and if it's near, that's no good because the image would focus behind the retina. That can't happen. So I have to put the opposite, a convex lens on there to bend the light first before it hits my cornea, then my lens, so that I get a clear image. Same with contact lenses. So contact lenses for nearsightedness will reduce that curvature of the cornea, causing light to focus directly on the lens. I'm going to skip this part on astigmatism. You can read through. The last bit are some common diseases of the eye, which you would, if you had one of these, you would go see an ophthalmologist. Cataracts are opacities. They're opaque portions, clouding of the lens. If you have that, you can get cataract surgery to replace the lens. Glaucoma, that's a buildup of pressure on the eye. That obviously, remember, any change in the geometry is going to affect how you see. So you actually can get severe enough that you have to drain fluid from the eye. You can also, if you play contact sports, football, rugby, things like that, a contact can actually detach the retina from the back of the eye. And of course, once again, that's a change in the geometry. It's going to cause problems. You'd have to go have surgery to reattach that. And a more common one, of course, is pink eye, and that's a actual bacterial infection of the eye. And you would need, so your eye turns bright red, and you would need antibiotics, usually antibiotic drops, to fix that problem. Okay, that's it for me. And once again, I know this wasn't the way we would have preferred to end the year, but if you followed through all the way, well, thank you for that. Hopefully you learned something. And we still actually have, even though this is the last formal day of classwork that I'm going to give, if you have any questions, don't hesitate to email me. I'm still, of course, checking email. We're still on the clock until the end of next week. So if you have questions, email me, and I will respond as soon as I can. Okay, that's it for me. And if you don't have questions, I hope you have a great summer, and hopefully I will see you next year at Beal. Okay, thanks, guys.